Good evening. Let's start a tradition for the class Immunology. Every Friday I want to do an Immunology Journal Club. And because we don't officially have class on Friday, I'm going to record it as a video and post it on YouTube so you can all see and answer questions about important papers with immunology. Let's start with week 1. This is a paper about macrophages. If you look at the title, I'm going to go into some of the details of the paper so that you can understand when you look at other papers and see details like this, understand how to process them. So let's start off with this one. Inhaled particulate accumulation with age impairs immune function and architecture in human lung lymph nodes. Let's look at the beginning and end of that. We're talking about inhaled particulates, and we're talking about their effects. Bad things happen in human lung lymph nodes, specifically with macrophages. So let's start with some of the background. I have to start with a little bit of chemistry and everything because I love the chemistry. And let's talk about combustion. Combustion, you're taught to it as a complete reaction where you have carbohydrates, for example, reacting with oxygen to completely produce water vapor and carbon dioxide. That's why we have so much carbon dioxide in the atmosphere right now. But incomplete combustion is a real-world kind of reflection of this. Sometimes the combustion is incomplete and you end up with carbon, just carbon by itself. And it's black sooty particles, and they disperse through the air, they get into the lungs, and they get into the body. So this is the chemical fact that we're starting with. The particles are non-self, and yet the body needs to discriminate them from self, and the body will sample all the stuff that gets inside the body, right? So the macrophages are going to try to eat some of these particles up. What happens when that happens, and when it happens over a lifetime? Incomplete combustion is related to why I suddenly had symptoms of asthma in my first couple days in Burundi. I was a little bit puzzled by this. Why usually going to a tropical area helps the asthma and wheezing, but then I was having a little bit of asthma and wheezing like I was 8 years old again. And then I realized it's because of a local industry. One of the things they do in Burundi is they have to basically make money and make things any way they can. One of the industries, if you want to call it that, is making charcoal. You basically take burnable material and you combust it into carbon. As you do that, you're doing open fires, and you have a lot of the soot that gets into the air. And I realized, after a little bit of trial and error experimentation with myself, I realized that it was these charcoal-making fires that was triggering my asthma. So you can see that those particles can cause reactions in the lung itself. Now we're talking not about short-term reactions in the lung, but long-term reactions in the lymph nodes. So before we talk about that, we haven't actually talked about lymph nodes that much. They're coming up in the class. But the whole idea is that lymph nodes are sort of a headquarters for the immune system, and they're connected to a separate set of vessels and organs known as the lymph. So you have blood and you have white blood cells that can go into the blood. But you also have a special transport system, a special kind of vessel for the immune system. So for instance, the innate immunity can occur when monocytes will actually be activated. They'll be turned into macrophages or dendritic cells. They will progress. You see number 2 right here is they will sample behind the epithelial barriers, like in the lungs. Macrophages will sample what comes through, and they'll basically just eat everything, and then they will carry it to the lymph nodes, where then the immune system decides what to do. The important thing for this is where the macrophages go to talk with the T cells and the B cells. There's some more details we're going to talk about when we get to it, but that's all you need to know for today. All you need to know is that there's these 3 types of cells. They all get together in the lymph nodes, and it's the macrophages bringing the information to the T's and B's. Now, the macrophages and dendritic cells are crucial elements to the T and B life cycle. We've mentioned this a little bit already, that dendritic cells are required for B cell activation. So in the lymph, you have certain areas for B cells and certain areas for T cells, and they are pretty much separate. Although the certain macrophages and dendritic cells will go to the different rooms, the different areas, when they need to do something, when they need to do a specific function. So all that is to say, all we care about right now is the macrophages can go to the T cell room or the B cell room. And it turns out that it has an effect on our story. So I want you to look at the abstract, and I'm just going to read it. And I want you to think about what stands out. What can you tell from just the abstract? Older people are particularly susceptible to infectious and neoplastic diseases of the lung. It is unclear how lifelong exposure to environmental pollutants affects respiratory immune function. In an analysis of human lymph nodes from 84 organ donors aged 11 to 93 years, we found a specific age-related decline in lung-associated but not gut-associated lymph node immune function, linked to the accumulation of inhaled atmospheric particulate matter. So I want you to think about what stands out from that. Pause and think about it for a minute. I have 3 major points that come from this that stand out that make this study special. So think about it. All right, here's my 3 points. This is a large study, 84 organ donors, and it's across a wide span of ages, from 11 to 93. And it turns out this was one of the largest studies of its kind. It was set up in 2011, specifically in New York City, and if you see the people who put this out, they're from Columbia University. That all makes sense. I noticed the distinction between the lung lymph nodes versus the gut lymph nodes. Usually people analyze blood because it's easy to get to. Because of their special setup, because of their special organ donor setup, they were able to analyze lymph nodes, and that makes this study special. Another thing that makes this study special is it has a very clear instigating event. The lung lymph nodes were literally darkened by the particles, and they showed this impaired immune function. And so we're going to talk about what does that mean. Impaired immune function has several parts to it. This is what they started with, and they must have just realized this when they were collecting all their lymph nodes, that the lymph nodes in the lung only turned black over age. These are 3 different donors on the left, showing the lung on the top and the gut on the bottom. And you see the gut on the bottom are sort of peach colored, right? The ones on the top, definitely there's something wrong with them. And it makes sense that soot particles could be somehow trapped in there. If you look at the lymph nodes from people aged 20, they are pink. These are the LLNs, the lung lymph nodes, but they are not black yet. You get all the way up to the dark-colored sooty ones, the ones that are clogged with particles. As you move up, they say that it's above 40 where they really start to become distinctly dark in most people. And they show you actually a whole bunch of different possibilities down here. If you look down on the bottom, we have lung on the left, we have gut on the right, and the lung ones, they have a measure of how many lymph nodes contain particles at all. And you see that there's a lot of scatter, but definitely as you go from 20 to 80 across, you're getting much more lymph nodes are containing the particulates. And so, if you compare it to the gut lymph nodes on the right, you can't see anything there. There's no particulate showing up in the gut. So this says something is going on, and the line we're going to draw is going to be 40 and older, or 65 and older. So the thing they found is the particulates actually become entrapped in a particular subset of macrophages. Now the name for these, they're named for the markers on their surface. Each marker is usually a protein that is on the surface. When this protein is on the surface, it plays a role in immune function. And it distinguishes. There are certain macrophages that have both CD68 and CD169. But then there are some that have one or the other. The macrophages that collect the particles have CD68 but not CD169. And they also checked, and they're not in dendritic cells, which have another set of CDs that are available. We'll talk about these CDs. You're going to see them in lots of papers, and so we're going to see them over and over again. Right now it's just a license plate. These are the CD68 positive macrophages. In fact, row 1 here, they show the whole lymph node here. The one way to look at it, look at the lymph node on the left. It has B cells are stained pink. And notice how all the B cells are sort of in these little nodules that are more toward the outside of the cell. These are called the follicles. These are the B cell rooms. The T cell rooms are more on the inside. And so one of the things they noticed is that if you look at the CD68 positive, CD169 negative, by the way, that's row 1. You see row 1 of the 3 rows. Row 2 and row 3 are different combinations of positive and negative. Okay? But if you look at the top of row 1, you can see that these don't come anywhere near the B cell related follicles. They show them in arrows, and we have to zoom in. This is a tiny figure, so we have to zoom in. And the thing about if you have the green C68 marker, you're going to be colored green. If you have the CD169, you'll be colored red. If you have both, you'll be colored sort of yellow with the two colors overlap. What they're saying is that we can see a certain number of cells, and these are the ones that have the particulates. They are only green. They don't have any red. They aren't yellow. They don't have both. And in fact, they have little arrows pointing at the little green dots, and they're saying, hey, look, these are where we saw particulates. These are where the CD68 positive, CD169 negative cells are located in the lymph. Definitely not in the particles. And that makes sense because these have a role to play with T cells. They did a graph for how many of these 3 kinds of cells. Again, you have positive 68 are the black dots that they show here. I guess they chose black dots because of the particulates to sort of represent that. And these are only in older patients, and as you see that the number of cells containing particulates, they were mostly showing up in the CD68 positive ones only. You can see how far, how much scatter there is, but it's definitely, I believe them, despite the scatter, I believe them that the CD68 positives, the black dots, are the ones that are always higher than the red dots and the red squares and the blue triangles, which are the other types of macrophages. So if you, and the other thing I noticed in looking at the data, notice that there's like 2 or 3 65-year-olds that have really high readings. I wonder about the life histories of these people. They were not smokers. They excluded smokers from the data set. Did they work around smokers? Did they live with smokers? Did they actually work in some kind of sooty kind of area? I don't know. But it would be an interesting thing to correlate with this. Here they do flow cytometry, and what this is just as a map, each dot is a cell. And they run the cells through the flow cytometer, and they're able to see how much of each CD is on the cell. And what they have is they have a graph here where they have lung lymph nodes on the left and gut lymph nodes on the right, like always with this. And what they have for this graph is on the y-axis you have CD68, on the x-axis you have CD169. And these are, by the way, are macrophages containing particles. Okay? So if you see it on this graph, that means it has a particle in it, and we wonder what kind of cell it is. Well, when they run it through, you see this quadrant right here in the lung is really increased. That means that the cells have gone up on the graph, meaning they have more CD68, but they have not gone to the right on the graph, meaning they don't have more CD169. So this is another example of a flow cytometry graph, the first of many, but I want to show you how to read it. You look over on the gut on the right, the same quadrant has hardly any CD68 positive cells. And they have the percentages in the corners there, so you can see that there's a lot of cells that aren't showing either of them that came through the flow cytometer. Those don't really matter. What matters is the percentage of cells that are in each quadrant, and the quadrant that we're looking at that's outlined in pink is 3.77. That's a lot more than 0.057, which is basically 0. So they tested the macrophages to see how they work. And they tested to see do the particles actually interfere with the macrophage function. And they found out they definitely do. There are 3 ways. They found they're not in an activated state as much as other macrophages. They found they don't produce as many cytokines. And they also said macrophages are going to phagocytose, right? These particle-containing ones don't phagocytose as well. They have significantly reduced because they're probably full. Basically, these macrophages look old and sick, and the macrophages themselves look aged. They also look stuffed full with particles. So those two things appear to go together. So let's go again and again with our CD68 positive macrophages. These are different CD markers, and again we have lung on the left, gut on the right. And the thing that this is arranged by age, and we have 3 different CDs that just show how healthy and active the cells are. And if you look in 20-year-olds, all of these markers are up in all the types of cells. If you go over to 80-year-olds, though, you see that the top 2 at least are dropping as you get older. In fact, some of them get down to 0. They lose these CD markers as CD80 and 86 and CD36. I do not expect you to remember those numbers, but I expect you to remember that 80-year-olds have fewer markers of activation on their specific macrophages than 20-year-olds do. And again, the really nice part of this experiment, you look in the gut, they're flat. The gut macrophages are not impaired in activation or in displaying these particular markers. So how much do they produce as far as pro-inflammatory cytokines? On the top, we have a comparison of macrophages with and without black particulates. The important thing is to see how things drop as you go from the red without black particulates to the blue with black particulates. They basically fed them particulates, and they watched the macrophages stop producing these cytokines. And these are the kind of cytokines the macrophages are supposed to produce them, so the immune system can make an inflammatory response. The immune system's not working right if these are going down. And you notice that there's not much difference for the younger people on the left side of each graph, but for each of these 3, TNF alpha, IFN alpha, and IL-6, as you go across from left to right, you can see that as you get to the older macrophages on the right, you get more of a drop. Now, not every macrophage drops, not every sample this is kind of messy, but you can see that overall you have the blue lines tend to be a lot lower than the red. You tend to have downward slopes as you're going from without to with, especially, and you tend to have more low blue dots when you move from below 39 to above 65. Now, you might look at this and say, well, the one on the bottom actually seems to be a mixed bag. It seems to be going all over the place, and that looks like it's not doing what you're saying. And the blue is not down relative to the red. And that's actually good, because the top 3 are pro-inflammatory cytokines. The bottom one's a control. That shouldn't change. It's just arginase, and it's just there on the surface of the cell. And they found out that it didn't really change. If anything, it went up in some cases. That shows us that's a nice control. So they actually did a test where they took their they took actually particles, and they fed them the macrophages. The macrophages would take them up. You can see under a microscope. If you don't have any particles around, they look nice and clear. With the particles around, the macrophages phagocytose them, and they become sooty themselves. They tested this under different temperatures. They basically tested it under body temperature. Only under body temperature do you actually get the cells able to phagocytose the way that they should. If you make them cold, they don't phagocytose right. If you add particulate, it's like you're making them cold. It's basically like you're putting them in the fridge. And so they just aren't working right. I want to show you the actual assay. There's a lot of details here you only need to know if you're actually growing macrophages. But I want you to see there's a recipe for doing it. You add penicillin, you add streptomycin, you add some glutamine, and you let them grow. Then you differentiate them using a particular molecule that turns them into macrophages. And then you add the particles. And this is something I found interesting. You buy the particles from Sigma. Just like everything else that we do, you can apparently, the Sigma will go and take atmospheric particles from air filters, and they're actually analyzed by the National Institute of Standards to be able to say these are good particles for your experiments. They're very homogeneous, and then they were purchased that way. So we can buy a bottle of particulates if we want to. Just look quickly over the rest of the kind of things. This is really cell biology. This is why we don't have a lab with this class, because it'd be very expensive, long, and what you learn is how to take care of cells. What you need to learn is the theory of immunity, and so that's what we'll talk about. And so if you look at the lymph nodes, let's go back and look at the lymph nodes and say, is there a physical change that you can see in older people that could be connected to this? And it turns out that the ones in the older people, especially the ones with particulates were close to disrupted B cell follicles. Look at the B cell follicles. They're really nice and circular in the 23-year-old and the 19-year-old. But as you move down, and LLNs, again, are on the left, gut are on the right. Very circular for young people in both lung and gut. As you move down to the 60-year-old and the 83-year-old, the ones on the left lose their circular behavior. It's a little bit hard to see because these are all a little bit hard to see, but I think even at low resolution you can see they aren't forming nice circles anymore. The B cell follicles look wrong. They look dispersed and not as good. And you can see the particulates in the lymph that are, looks like it's associated with that. They did a study with a lot more of these than they can show, and they showed that overall there's a significant association between the particulates and disrupted B cell follicles. So, I want to read you quotes from the paper's discussion section. I want you to hear what they say, and I want you to think about what these mean. Diseases of the lungs and respiratory tract disproportionately affect older people, as has been evidenced by the dramatically increased susceptibility to respiratory infections that has been observed during the SARS-CoV-2 pandemic. This is like saying SARS-CoV-2 was worse for older people because it was respiratory, in part. And it's not because of some specific problem with the virus. It was a bad, previously not seen virus, but the fact that it was respiratory meant that it hit older people harder, just as a nature of it. So here we reveal a new mechanism for compromised respiratory immunity with increasing age. This might be causal, okay? So our findings provide evidence for individual and cumulative effects of environmental insults and senescent changes on lung-localized immunity. Basically, I like environmental insults. I think it really says what's going on. And the important thing, we provide direct evidence that macrophages take up atmospheric particulates that lodge in LLNs. They lodge in lung lymph nodes. And so why do they lodge? We don't really know yet, but they're saying, I think this is pretty good evidence that they lodge and that they're bad. They're causing bad things. They're not just hanging out and doing nothing. So macrophages in the lymph nodes of younger individuals exhibited higher turnover than those from older individuals, which may facilitate particulate clearance. In other words, younger people can flush out and make new macrophages more quickly. The old macrophages get removed. Why do these older macrophages stick around and get so dark that they turn the lymph nodes black? We don't know, but that's what I'm sure they're working on. Here's another thing from their previous studies of age and immunity. They showed that proportions of tissue-resident memory T cells and influenza virus-specific tissue-resident memory T cells were maintained over age in human lungs, but decreased in frequency in the lymph nodes, suggesting that altered lymph node architecture also impacts the maintenance of memory T cells. Basically, memory T cells stay the same in lungs in older people, but where they go down is they go down in the lymph nodes. Something going on in the lymph nodes is impacting the number of memory T cells. Those are the ones that remember old diseases, and it might be why immune systems in older people have so much trouble. So, if you want a quick summary of this paper, you are what you breathe. If you breathe uncombusted carbon, you will eventually, that uncombusted carbon will become part of you and will interfere with your systems, specifically your immune system and specifically your lymph nodes. not the immune system in the lungs. This is very interesting. If you've heard of the ship of Theseus example, this is kind of like it, in the sense that young macrophages are cleared out more frequently, and that's like the ship of Theseus, where it's able to get replaced. You can look it up if you need to. If you haven't heard of it before, okay, it's not crucial to this. But the immune system itself seems to age over time, because if you can renew your cells, you can stay young, but you can't renew your cells, especially if you have environmental insults. We already knew that older people had fewer naive T cells and lower thymic output. This is one of the things I found out when I was curious as to why so many older people were being hit so much harder by COVID. Turns out that their T cells, they don't have as many. Their memory T cells are not maintained. And they have lower thymic output. We'll explain that when we get to the thymus. But it turns out that what's true for T cells is also true for macrophages. Their macrophages, rather than having fewer young macrophages, they have more old macrophages. Their macrophages are clogged with particles. They have B cell follicles, and it's not just the T cell areas of the lymph node that are affected, also the B cell follicles, where these macrophages don't even go, that we can know. They also get disrupted. So it looks like this is one of the ways in which people age. And there's actually a little bit of hope. If there's a specific subset of cells, the CD68 positive, CD169 negative macrophages, if they are the ones that have the problem, maybe we can solve the problem by focusing on it and coming up with a specific therapy to either clean them out or renew them and replace them with other cells so that they aren't hanging around with all of these particulates. At any rate, well, thanks for listening to the First Journal Club. I am not quite sure how long this was, but I want to take the time we need to go through the paper. 25 minutes, okay, I'll take that, less than half an hour. I will see you on Monday, and on Monday we will have a quiz that will include some information from this. Hopefully you've been taking notes, because you can use those notes on the quiz. All right, see you Monday.